Welcome in learning immunodiffusion. Immunodiffusion is a technique to detect or measure the antibodies and antigens by their precipitation when diffused through a gel or other medium. We will learn in detail about this technique under the following learning objectives, which include knowing about its principle, different types, procedure, application, and finally we will conclude with a brief summary. Moving on to the principle. When a soluble antigen combines with its antibody in the presence of electrolytes, say sodium chloride, at suitable temperature and pH, the antigen-antibody complex will be formed. The formed antigen-antibody complex forms an insoluble, visible, distinct band of precipitation in the soft gel agar or agarose medium used in the technique. This is the basic principle of the technique. Know that there are 4 different modifications which include single diffusion in one dimension, also called as Ordin procedure, double diffusion in one dimension, also called as Oakley-Fulthorpe procedure, single diffusion in two dimension, also called as radial immunodiffusion or Mancini single diffusion, double diffusion in two dimension, also called as Octerney double diffusion. Out of this 4 different modification, we will know in detail about the last 2 mentioned modification in the slide, as they are widely applied in the diagnostic. Knowing about radial immunodiffusion, discovered by a scientist Mancini, named after him as Mancini single diffusion. In this diffusion, the antibody will be incorporated into the agarose gel at first. Once the incorporation is done, the agarose gel will be poured on a flat surface. The flat surface used here can either be a slide or Petri dish. Once the agarose gel solidifies on the slide, the wells are cut using the gel puncture and the antigen will be loaded in the wells. After loading the antigen will diffuse radially from the well and it forms a ring-shaped band of precipitation called as precipitin ring. Once the precipitin ring is developed, the concentration of unknown antigen can be measured by comparing it to the standard antigen and using a standard graph. A standard graph of diameter of ring on the y-axis versus concentration of antigen on the x-axis is plotted using a semi-log graph, and the concentration can be determined by noting down the diameter of the ring. Procedure of single radial immunodiffusion performed by a kit method in detail ahead. Components of the kit include agarose, antiserum, and 10x assay buffer. There are 4 standard antigens and 2 test antigens. There's one gel puncture and a template to make the well on the slide. At first, the slide is used, cleaned with the alcohol. Agarose is weighed and transferred into a flask. Step 3 involves pipetting out 10x assay buffer and water and adding the agarose into the flask. The agarose is made to dissolve by keeping it in boiling water bath. Once the agarose is dissolved, the flask is taken out and cooled to 55 degrees Celsius. Step 6 involves adding the antiserum and mixing well in the agarose gel. Step 7 says pour the agarose onto the slide carefully without spilling. Then once it is done, the slides are allowed to solidify. Once solidification is done, the wells are made using the gel puncture according to the template given. Add the antigen into the respective wells and incubate the slides overnight with cotton. In incubation, you will see the result like this, wherein a clear precipitin band will be developed with respect to each antigen used. Here you can see 6 different antigens, out of which 4 antigens are standard antigens and the 5th and the 6th antigens are the test antigens, wherein the concentration of those is unknown. So suppose if you want to find the concentration of the unknown, at first you need to take a scale and measure the diameter of unknown antigen. Once you have noted the diameter of unknown antigen, plot a semi-log graph. Here a semi-log graph is used because the concentration of antigen increases exponentially. You can see here in the graph the concentration of antigen in milligram per ml is plotted on the x-axis and diameter in centimeter is plotted on y-axis. Say the constant diameter of unknown antigen used is 0.6 centimeter. The concentration of antigen opposite to it will be 0.3 milligram per ml. This is how, using a standard semi-log graph, the concentration of antigen can be determined. The application part. The technique can be used to determine the relative concentration of the antigens and antibodies, that is by radial immunodiffusion method. Octernally double diffusion method can be used to compare different antigens and antibodies as discussed. And then the relative purity of the antigen prepared can be assessed by having the standard antigen, and the different class of immunoglobulins in serum can be analyzed, and also the antibodies to influenza virus can be screened using immunodiffusion technique. Diphtheria, which is an acute pharyngeal or cutaneous infection, and the toxigenicity of this bacteria can be screened using immunodiffusion. In forensic application, the blood and the seminal stains can be identified. Food adulterants can be tested, and the method can be used to standardize toxins and toxoids. The VDRL test for syphilis, that is Venereal Disease Research Laboratory test, uses the principle of immunodiffusion. Summarize, we learned about what is immunodiffusion, what is the principle of precipitation, what are the different modifications of the method, out of which single radial immunodiffusion and octernally double diffusion was learned in detail, looked into the detailed procedure of radial immunodiffusion, and also saw the application of the technique. Hope you enjoyed learning. Thank you.